emotions is part 2 of this. So first we're going to talk about hormones and reproduction. Okay? And what are hormones? We already talked about this, right? What are hormones? Chemicals released into your blood supply, right? Vasopressin, that was a hormone we talked about, right? It causes water conservation and blood vessel constriction. So a hormone is any chemical secreted by a gland or released from a neuron that's carried through the blood. That's the key thing, it's carried through the blood. And its target can be another gland, like the adrenal gland, for water conservation, or the kidneys, an organ, or it can be a neuron. Hormones are good because they have widespread effects. Everywhere the blood goes, the hormone goes. They're good for sustained effects, long-term effects, because they don't go away very quickly because they're circulating in your blood. Right? So neurotransmitters have very localized effects at a synapse, and they're very short effects. Hormones have long, sustained effects. There are 2 general classes of hormones. Most of the hormones are what we call peptide hormones, and these bind to receptors outside of cells to cause their response, like a metabotropic receptor. There are also steroid hormones. And so the sex hormones that we talk about today, a lot of those are steroid hormones. The difference is the steroid hormones can actually go inside of the cell. They can pass through that plasma membrane, through the lipid, and have effects on the nucleus and other things inside the cell. Sex hormones, as I mentioned, are a good example of steroid hormones, like testosterone, which is not up here, but estrogens, progesterone. Testosterone is a type of androgen. All right, so peptide binds on the outside, causes second messenger to be released, has an effect on cell excitability. Steroid hormone passes through the lipid membrane and can have direct effects on the cell, inside the cell. Good? Good. The endocrine system, we've already talked a little bit about this. What's the master gland? What do we call the master gland? The pituitary gland, right? So neurons in the hypothalamus go down and innervate the pituitary gland, which is the master gland, and we call it the master gland because it often releases hormones that go turn on or off other glands, other parts of the endocrine system. Okay? So it's like the puppeteer controlling the other endocrine system glands in the body. And that can be things like the liver, the pancreas that we already talked about with insulin and glycogen, glucagon, insulin and glucagon, the thyroid gland, and then we'll talk about the ovaries and testes today through reproduction. The pituitary gland is separated into 2 parts, the anterior and the posterior. And it's outside the blood-brain barrier. What's the pituitary stalk called? The infundibulum, right? And so this is outside the blood-brain barrier, so it can release directly in the blood and have hormones, right? The things that you need to know about the anterior and the pituitary, there are 2, the anterior and the posterior pituitary, are that they release different hormones, and they release them in a different way. Okay? And the easiest way, I think, to learn this is to focus on the posterior pituitary first. Because the posterior pituitary only releases 2 hormones. The anterior pituitary releases a lot. So all you have to know are the 2 that are released from the posterior, and then anything else you see that's released by the pituitary comes out of the anterior. Okay? That's the easiest way to learn. The posterior pituitary, not only does it release only 2 hormones, it does so in a different way than the anterior pituitary. So the neurons from the hypothalamus that come down through the infundibulum to the posterior pituitary form synapses on capillaries, blood capillaries, and actually release their neurotransmitter as hormones directly in the blood. And those 2 neurohormones, neurotransmitter hormones that are released, are oxytocin and vasopressin. And as we mentioned, we already talked about vasopressin, we talked about fluid balance and temperature regulation, and we talked about vasopressin constricts blood vessels, causes us to conserve water in the kidneys. So antidiuretic and vasoconstriction. So there are vasopressin neurons in the PBN that send their axons down here and directly release vasopressin into the blood. There are oxytocin neurons, and oxytocin is released in response to different sexual behaviors and parental behaviors. And we also think that oxytocin is released in response to bonding. And that bonding can be monogamous male-female intimacy bonding, social bonding, love. Right? And it's also bonding that happens between a mother and her infant in terms of mother-infant bonding. We know, for instance, that there's big releases of oxytocin when mothers breastfeed, and that we think that that helps facilitate a bond, a social bond between the mother and the infant. We're going to start talking about some systems level stuff, love, attachment. These things are hard. There isn't a love nucleus in the brain. These are widely dispersed things that are difficult to study from a neuroscience level. What we know are bits and pieces like, hey, oxytocin goes way up when mothers breastfeed. It looks like that has a bonding thing. We know things like there's a vole, a mole-like animal, and there are 2 different strains or species of it. One is monogamous, finds a mate for life. The other one is polyamorous, will mate with all sorts of different ones. And the differences happen to be with oxytocin and vasopressin. So we think something about mating, monogamy, has something to do with this. But these are big things that have lots of other things involved. Right? All right, posterior pituitary, what do you need to know? Oxytocin, vasopressin, released by neurons directly in the blood supply. Anterior pituitary. Here, it's different. We have neurons from the hypothalamus that stimulate groups of cells, glands essentially, that those groups of cells then release the hormones. So, like I said, there's lots of hormones released from the anterior pituitary. Don't try and memorize all the hormones that are released from the anterior pituitary. Just memorize oxytocin and vasopressin from the posterior, and everything else you see, you know has to be anterior. Good? So, growth hormone, there isn't a neuron in the hypothalamus that releases growth hormone. There's a neuron that stimulates cells that then release growth hormone. So it's like a 2-step process, as opposed to the posterior, where the neurons themselves are releasing the growth hormone. Good? Questions? So then all of these different ones, we'll talk about ACTH tomorrow morning. That's the stress hormone. Growth hormone. Today we're going to talk about follicle-stimulating and luteinizing hormone in terms of the female reproductive cycle. Prolactin stimulates milk production during breastfeeding. Thyroid-stimulating hormone. When did we talk about thyroid? Basal metabolism, right? We talk about changing your temperature regulation. Basal metabolism happens through the thyroid. Right? Lots of hormones that come out. Don't need to memorize them. I'm going to use in order to understand how the brain and the endocrine system control processes in our body, we're going to look at two models. We're going to look at stress tomorrow, and ACTH and the adrenal cortex and cortisol, stress hormones. And today, we're going to look at the female reproductive cycle, the menstrual cycle, and pregnancy, and how the brain helps control the ovaries and reproductive system, and how the reproductive system then talks back to the brain through the release of these hormones as a model for this. So there's 4 chemicals you need to know their names and their roles in this process. FSH, follicle-stimulating hormone, is released by the anterior pituitary. That's why it's in green. Luteinizing hormone, also in green, because it's also released by the anterior pituitary, LH. So FSH and LH, anterior pituitary hormones release. Targets are reproductive organs. Estradiol, in blue, because it's released by the peripheral endocrine system as a signal back to the brain. It's a form of estrogen, those steroid hormones. It's released by the follicle and it sends a signal back to the hypothalamus. Progesterone, in red, also a peripheral signal, but for a different reason than estrogen, why it's not blue, but released from the uterine lining and sends a signal back to the hypothalamus, to the brain. Good? Now we're going to talk about how these things work together. So, at the beginning of the menstrual cycle, the anterior pituitary starts to release follicle-stimulating hormone, which, as the name suggests, stimulates growth of the follicle. All right? Nothing tricky there. Okay? And it nurtures the development of the ovum. So here we have the follicle that's developing with the ovum over the course of the first half of that menstrual cycle. As the follicle starts to mature, it's getting bigger and maturing, the follicle starts releasing estradiol. And estradiol then is a signal back to the hypothalamus that, hey, that follicle stimulating hormone is working, I'm getting bigger, I'm maturing, and so this signal is going up. Estrogen is going up. FSH is high, estrogen is going up. When the ovum is mature and done growing and ready, at optimal time for fertilization, this level that it's now producing of estradiol triggers a response from the hypothalamus to release a surge of luteinizing hormone. Luteinizing hormone, so the estradiol then triggers luteinizing hormone, and luteinizing hormone is now going to trigger the release of the ovum by the follicle, which is also called ovulation. And it's the maximum time of fertility. So you have to, there's this interplay back and forth. Hypothalamus releases FSH, follicle and ovum mature, releasing more and more estradiol till it triggers luteinizing hormone release, which then causes release of the ovum at the point of ovulation. All right. So, in addition to these sort of simplistic, like regulatory back and forth pathways, there's also complex influences of these hormones on behavior. So, since ovulation is the maximum time of fertility, it also, these changes in hormones also trigger changes in sexual behavior, and reproductive behavior, and desire. And so this is looking at women that are taking birth control pills in blue, women that are using intrusive methods of birth control like IUDs, and women that are using non-intrusive methods or not on any birth control. And what we see is if we're recording their female-initiated heterosexual activities, at the period of ovulation, that goes way up for the red group. They are now initiating more heterosexual activities at the point at which they're most likely to get pregnant and most fertility because of complex actions of these hormones in those reproductive areas of the brain, those reproductive behaviors in the brain. Right? Not pheromones, but these are internal signals. Right? If we look at the blue line, what happens?
It dips.
It dips a little, but it pretty much stays kind of flat. I mean, it doesn't dip a little, but if you look at the variability there, it's just bouncing around in this level. Not up here, but bounce around this level. So chemical birth controls work by tricking the body into thinking that it's pregnant and not ovulating. So you don't get these changes in luteinizing hormone and estradiol because of the chemical birth control, and therefore you don't see the increases in sexual activity that are associated with those chemicals. Okay? Here you see a little bit of a bump with the intrusive methods because you're not interfering with the chemical balance, but not as much as those that are not using any of the green lines. Right? The bottom line, you kind of ignore the green line and think about the blue. Blue, no chemical signal. Red, chemical signal. All right. After ovulation, the follicle now starts releasing a chemical called progesterone, which is going to cause the uterine lining to become thicker, and allow an ovum that's been fertilized at ovulation to implant and grow as an embryo. If that ovum is not fertilized and there isn't an embryo to implant, right, then those levels will decrease and you'll have your menstruation period where you'll shed that buildup of the lining, and then the whole thing starts over again from baseline. With, what's the start of it? What happens at the start of the cycle?
So it reduces FSH.
The hypothalamus causes pituitary release of FSH. So, I'm sorry, I should mention. This progesterone, as it's not only thickening the uterine lining, is inhibiting the release of FSH and LH. So they stay very low until it goes away at menstruation. Once it goes away, these are no longer inhibited. That's why they can become active. Sort of forgot. Okay. If, as I mentioned, there's pregnancy, the ovum does become fertilized in ovulation and implants, then those levels of progesterone and estrogen remain high throughout pregnancy, which prevents LH and FSH from being released until the pregnancy's over. And so you don't have menstrual cycles during pregnancy because you're preventing those chemicals from being released. And this is essentially what chemical birth control pills do. They trick your body into thinking they're in this state to prevent the release of LH and FSH, so you don't get a follicle stimulated, and you don't get an ovum released, and you're not subject to pregnancy because you don't have those things available. All right. this is from your book, right? And I like this because it sort of shows the communication system back and forth. Starts with FSH, causing the follicle to grow. The follicle, as it grows, releases more and more estrogen until it triggers that surge in LH and FSH for ovulation. Then the follicle then inhibits the release of FSH and LH until such time as it's shed or the pregnancy's over. Good summary? All right. Since we're talking about reproduction, we'll talk a little bit about sex and sexual development. I'm not going to get too much into all the clinical sorts of abnormalities of that, but we'll talk a little bit about it. So, in terms of, most of you will probably consider having children at some point in your lives, maybe. You're a sucker for punishment. Says the dad of 2 teenagers. Love them both dearly. So my question to you is, can you choose the sex of your child?
I mean, you can't biologically, but now you can actually change.
Okay, so biologically you can't, but you can manipulate things genetically, right? You guys have grown up in a different time. I think this is maybe the first class that's really caught on to that. Most are like, no, you can't do that.
Can you fully change the sex though? Can you fully change the chromosome? I know you can change parts of it. I don't
You can, we're not talking about changing, we're talking about choosing. We're not talking about taking a male, making a female, or vice versa. Although you could do that very early on. So what determines the sex of a child? X and Y chromosomes, and you get one contribution from the egg and one contribution from the sperm, and how they recombinate. If it's XX, comes female. If it's XY, comes male. Right? We've already talked a lot about sex chromosomes and sex-linked disorders, where males may be more susceptible to mutations on the Y chromosome because they don't have, or on the X chromosome because they don't have a backup one. So if they have a mutation here, and females have a mutation here, they have a backup that can take over for that mutation where males don't, like color blindness. All right, so we know one of each of these is contributed by a sperm and by the egg. What do we know about sperm? Well, what do we know about the, right? It's the sperm that contributes the X or the Y. We have Y sperm and X sperm. The female contributes the X. And what we know is that sperm that contribute the Y chromosome are different than sperm that contribute the X chromosome. Y chromosome sperm are smaller, they swim faster, they also die quicker, they have a shorter survival time, and they're weaker. And all the women in the room say, I knew, man. Die sooner and are weaker. They may be faster, but X sperm are larger in size. They swim slower, but they have a longer survival time. They're more robust, particularly in different acidic environments, pH or environments. So we can use this information, right, to your benefit if you want to bias the system towards having a male or a female. It doesn't mean you can choose your sex, but knowing and understanding physiological differences, there are things that you can do in reproduction to bias the Y sperm having better access to ovulating egg versus the X sperm having better access to an ovulating. Also, this difference in size means that we can do things like in a lab, spin them so that the larger sperm are in one part of the test tube and the smaller sperm are in another, and separate them. And so in artificial insemination, you can bias it towards one sex or the other. So when we're talking about in a laboratory choosing that, right? And beyond just artificial insemination, and we do sperm filtration based on size and pH. And in addition to artificial insemination, only trying to inseminate with X or Y sperm, if you're doing in vitro fertilization, IVF, there you're actually combining the sperm and egg in a dish, a culture dish, and then implanting that embryo. And so you can choose to only implant embryos that have been created with the X or Y sperm, right? So there are ways through reproductive, I'm not trying to say, fertility treatments, sorry, fertility treatments that you can bias it one way or another if you would prefer to have a boy or a girl. It's not 100% effective, but you can definitely change the odds. You can also change the odds in normal reproductive behavior depending on the placement and the timing of the placement of the sperm. Because remember that the male sperms are going to swim faster, so the closer in time you are to the ovulation, to an egg being there ready, right, then you're biasing that to Y sperm getting there and having more availability to impregnate the egg than X sperm. And the closer that the sperm are placed towards the egg, right, then you're also biasing the Y sperm to get there and have more selection. The further away, the further away you are from placement of the sperm, then it's going to take longer to travel there. So therefore, more X sperm are going to make it because they can go longer distances, survive longer, and less Y sperm are going to end up making it. So where the placement sperm is in reproduction can also bias towards having a boy, right? We already talked about that. You can also, there's other sort of more complicated ways that you can talk about pH environments of the uterus and things like that to, again, bias it towards one or the other. All right. So a lot of people, fertility treatments have become much, much more commonplace now. And it's probably important that you understand a little bit about them. And how many of you know someone personally that's gone through fertility treatments? Right? It's about half the class, so it's pretty common. That wasn't always the case. Right? So how many of you know how much it costs for a cycle of artificial insemination? Anyone have a guess?
Maybe 5,000, depending on how hard your process is.
Yeah, it's pretty expensive. It's anywhere from $300 to $4,000. It's a ballpark, right? But it's not cheap. And it usually takes multiple rounds, and it usually doesn't work the first time, right? So every round, it costs that much. So it can get expensive quickly. There are two different types of artificial or fertility treatments. There's intrauterine insemination, which is where we're now going to be placing sperm directly into the uterus, right, to allow sort of a normal fertilization process within the uterus. Or there's, as I mentioned, IVF, which is where we're going to do the fertilization in vitro outside the body and then implant fertilized embryos into the uterus. Usually takes 3 to 4 cycles on average to be successful. I'm sure you probably have heard horror stories of 10, 12, lots of cycles, people giving up because it's not happening, and so it ranges. When you're trying to select for sex, as I mentioned, it's not 100%, and some people don't care, they don't want to do that, they want it random, which is fun. But when trying to select, there's about an 80% success rate for both, 83, 80, 78. So you can do these things to sort of select for the sex of your child. As I mentioned, naturally, there's a lot of books out there and websites, there's thousands of websites probably, a lot of misinformation and myths and propagandas But if you understand the science of sperm placement, timing in regard to how close or far away you are from ovulation, pH environment, you can sort of bias things one way or another if you're particularly interested in having a word. Why would people want to do that? Do you think that's normal, abnormal? Why would you think somebody might want to try and bias towards having a particular sex as a child?
Well, for those who want to carry on their last name, males, they have more of like, I don't know, the stereotypical thing where guys want to carry on their family name, and
Okay.
so they tend to want boys so they can.
Having had both a boy and a girl, there's differences in raising boys and girls and pluses and minuses of the things you get out of both of those. And you think about people that have had 4 boys and they really want to have a girl. I mean, there's all sorts of things, reasons that people might want to do this that are, their own choices, right? All right. So a little bit about sexual development. So we talk about X and Y determining male and female sex. So the gonads, the reproductive tissue, the ovaries and the testes are derived from the same tissue developmentally. They start out as the same tissue. And then the presence of particularly the Y chromosome causes a change. So again, women, you are the default choice. And so not having a Y chromosome, or in some of the genetic disorders where people actually have a Y chromosome and they don't produce testosterone, there's a mutation in the SRY gene that don't produce testosterone, then the body will develop the reproductive system of a female. So it's the presence of a gene, SRY, on the Y chromosome that causes the gonads to develop into testes. And once the gonads develop into testes instead of ovaries, they start to produce testosterone, which then masculizes, changes all of the reproductive organs into the male reproductive organs versus the female. So it's really the presence of this gene on the Y chromosome that causes testosterone that causes these changes. Mutation. And we're back. So, couple of myths, right? Men and women have very, very similar cognitive abilities, okay? The variance in cognitive abilities across men and the variance of cognitive abilities across women overlap so much that we can't say that there's any difference between men and women in their ability to do math, to be creative, to be an engineer, to be a musician. There's none like that. And I say that because for a long time, some people, probably with some political motives, perpetuated this, that men were better at things than women, and women might be better at other things, and to perpetuate stereotypes and other things. And we know the science on that is pretty definitive. That yes, there's some men that are really good at engineering. There's some women that are really good at engineering. There's some men that really are good at engineering. And the variance is very big, right? And there's literally no effect of sex on cognitive ability. I think it's clear. Men and women's brains are not different sizes. Different ethnicities don't have different brain sizes. These are all things that have at one point or another been put forth by people, and they're just not true. Okay? But what is true is that men and women's brains are different in some very specific ways, right? So testosterone causes organizational changes in very specific places in the brain. Can anyone think of a place in the brain that would be different between men and women that we've talked about today in this lecture? Pituitary. Not the pituitary, that's not in the brain, that's a gland. close. Do men release luteinizing hormone and follicle stimulating hormone? Do they have a hypothalamus area that's in charge of detecting estradiol and progesterone in the reproductive cycle we just talked about? They do not. That is a gender-specific, not gender, a sex-specific difference in male and female brains, right? So there are some very specific differences related to males and females, right? One example we already talked about, right? There's also some other parts of the brain that structurally we can detect differences, but we don't know that because one area of the brain is bigger or smaller, that there's a specific functional significance to that. Right? So it's a big jump to say this area is bigger or smaller to say what it means behaviorally or functionally. But we can measure differences. So as I mentioned, these are descriptive, and it's really hard. You're making a lot of assumptions to jump to functional differences. Okay? And then I mentioned gender, but gender identity is the gender with which one associates themselves. And it's separate from sex. Sex has to do with XY chromosome and development of the body versus your identity with the sex, your association with the sex is gender. Okay. So Simon LeVay, famous name. You will see this on Psych GRE. You will see it on your exit exam when you're a senior. There's a couple of names in neuroscience. Remember I had that sheet at the top of the page. This is one of them. And he is known for sort of really pioneering studies of a part of the hypothalamus called the anterior hypothalamus, actually called interstitial nucleus number 3 of the anterior hypothalamus, that when he measures that, and so here he's measuring it as smaller, here he's measuring it as bigger. Do you see the border around the nucleus in either of those? The wall? No, you don't. There is not. It's a little subjective. It's where are those nuclei closest together? What are their connections? Why is this neuron not part of this nucleus, but this neuron is? Right? There's a lot of variances, right? But in general, bigger, smaller. And in looking at women and men, in females, this nucleus tends to be smaller, more compact, and in men, it tends to be larger. Not in all females. Some females, it looks just like a man. Not in all men. Some men, it looks just as small as a woman. But in general, statistically, if we look at this, there's a difference. Okay? So, in animals, so what does this do? That's a descriptive difference. Well, if we want to know what something does, we can go in and lesion it and see what changes in the animal. And in animals, if you go in and lesion this area, they lose interest in sexual reproduction. It has to do with sexual behavior. Okay? And the sort of controversial thing at the time that I hope is not as controversial now is that not only did Simon LeVay look at heterosexual men and women, he also looked at homosexual men and women. And what he found was that in homosexual men, their interstitial nucleus tended to be, on average, much smaller, like female. And when he looked at, I don't have that data here, homosexual women, it tended to err on the much bigger side, right? The importance of Simon LeVay in this study is it is the first time that we were able to demonstrate a biological difference that could underlie sexual orientation, whether or not you were attracted to men or attracted to women sexually. And it was the first time that homosexuality was sort of validated as not just a behavioral choice of an individual, but a physiological thing that could exist that could drive that change. Now, it's not the only thing, okay, here are, in this sample, two heterosexual women that have just as big nucleus as men, but they are not homosexual. And here we have heterosexual males that are not homosexual that have that same size as the homosexual. So it's not like the size of this nucleus determines your sexuality, but it clearly has some sort of complex interaction, and clearly there's some biological influences on this determination. If you ask homosexual people, about, their gender identity or their sexual orientation, they'll tell you, I don't know, I just always felt that way. It's just a natural thing. It wasn't like I woke up one day and decided I'm no longer heterosexual, I'm homosexual, right? And they rarely ever report that. All right. So, in terms of sexual orientation, if we look at siblings and we look at monozygotic twins that share the same genetic background, dizygotic twins that share the same uterine environment but different genetic background, so fraternal twins, identical twins, and adopted brothers, adopted siblings, same familial environment, different genetic background. Or here's a non-twin sister. This would be a sibling. I don't know why they don't have a sibling name, right? The concordance rates there, when the genes are identical, 50% of the time, if one twin has homosexual orientation, the other twin does. Compared to only 22% shared environment or 11% adopted. So is there an influence of genetics underlying this? And clearly there is. Does your genes determine your sexual orientation? No. 50% is not 100%. So 50% of the time, the identical twin of someone who reports as homosexual is heterosexual. Right? But clearly there's an influence, a biological influence, on these complex human behaviors. Right? All right. So, in terms of sexual orientation, preliminary reports, these are things that are descriptive correlations. They're anatomical things. It's hard to assign a functional significance to them. We're looking at correlations between people's behavior, sizes of areas of the brain, or genetic background. Just correlation, not causation, right? And we don't know what are the influences. We already know that environment, through epigenetics, that we'll talk about tomorrow, has strong influences on genetics. Right? All right. One last thing on this topic before we switch gears and talk briefly about emotion. On pages 459 to 461 in your book, there's a section on social neuroscience. I got nothing against social neuroscience. Very interesting, newer field of neuroscience, looking at these complex things like sexual orientation, gender identity, love, empathy, feelings that are very complex, right? How do we have these come from the brain? Let's figure out how they work, right? But they're really complicated. And sometimes people try to take more simplistic analyses and apply them. So for instance, there's a study in your brain, in your book called Love in the Brain Study. It was an fMRI analysis, and essentially they put people on an fMRI, showed them pictures of strangers, took pictures of their brain, showed them pictures of their loved one, their significant other, took pictures of the brain. Some areas of the brain lit up differently when they saw their loved one compared to strangers. And then those researchers said, here, look, this is where love is in the brain. These areas that got active, that's love. No, it's not. So you can think about all the confounds that would be in that experiment, right? When you see a picture of your significant other, you think about a lot of different things. You have a lot of different emotions than if you just see a picture of a stranger, right? So it's not necessarily where love is in the brain, but that was their conclusion, and it got picked up by newspapers and TVs, and they're like, we found love. No, it's not the way it works, right? It's very important design, by the way. Oxytocin, as I mentioned, in an animal model of a vole, the prairie vole and mountain vole, shows that oxytocin is the thing that causes those voles to be monogamous, presumably form social bonding and trust. And when it's not there, they're the non-monogamous version of the vole, right? Well, again, and there's been some studies where people have done things like spray oxytocin up into your nasal cavity, because it can be just like vasopressin, picked up and transported to the brain, and then given people experiments where they test their trust. So they're like, how much are you willing to gamble that this, on this person? Do you trust that this person as your best friend? Like they try to develop methods that would get at trust. That's a difficult thing to do on its own. How do you measure if someone trusts someone or not, right? Very difficult. But these studies showed that when they got oxytocin, they tended to trust the other person more. So then this became published, and all the newspapers picked it up, and TV, picked it up, and news media, and it went through the whole news cycle, and they're like, hey, we found the trust hormone, oxytocin. And then you get, and so as I mentioned, it's identified here, it's correlated with parental behaviors, as I mentioned, bonding with mothers. Then you get companies like Liquid Trust that are now selling what they call is oxytocin in a bottle, right? And they're like, spend $50 on this, put it on in the morning, and then everybody you interact with will trust you. Selling cars, when people will trust you, wear oxytocin, they'll trust you. You'll be able to sell more cars, right? Just not the way science works. What are the problems you see with this? I mean, they're sitting here they're saying, here, spray directly on your clothing. You will have more confidence, more intimacy, stronger bonds, right? It's not for our newsletter. And then here's the kicker, though. The science, human connection. So they have.
What does the quote say down there?
For pets.
What quote?
Right above the, keep going, right there. They swear by these products. Sometimes I don't know if my family or my mind eats them more.
So maybe they had to take this off, but they used to have a big, I haven't been here in a while, they even have it for pets, good lord. So, anyway, they used to have a whole section on here as seen on BBC, ABC News, CNN, as published in Nature Publishing Journal. And all of that's talking about this actual real study that came out. But what did they do differently in the study than they're suggesting here? How did they apply the oxytocin in the study? Spraying it up your nose, right? And they're saying, all right, right away we're saying, okay, you just put this on your clothes and it will somehow waft over to other people in sufficient quantities to get up in their nose. And this isn't the coronavirus. You got to spray this stuff up in your nose, a lot of it, in order to generate these effects. So right away there's an issue there. Never mind the ethics of, do you really want to meet your significant other because you wore oxytocin and tricked them into trusting you? It's just, okay. Anyway, I should go. Should we give up? No, right? But you have to be very wary of these studies that make big, broad conclusions about these really complicated things that are difficult to study, but we should study them, right? All right, I'm going to give you guys a reprieve and not make you do your jamming now, because we're going to get through this and be done. All right? Emotion. So that's reproduction. Good? Bueno? Interesting, boring, already knew it all. Okay. Emotion, right? So emotions exist on a continuum. So you can have low emotion, valence, like I'm annoyed, I'm happy, I'm apprehensive. And then if we take happiness and we ramp it up, you can have joy at a more intensity, right? Joy is a more intense form of happiness. Anger is a more intense form of annoyance. I was thinking arrogance, of annoyance. And then you can have even higher levels like rage or ecstasy, right? You can have apprehension, fear, or terror, right? So there's some basic 8 sort of people that study emotion come up with 8 universal emotions that all humans show and interpret, irregardless of culture, geographical location, indigenous peoples in the South Pacific to Canadians, all show the same 8 emotions. Doesn't matter your culture, where you grew up, right?
Sociopaths? Even sociopaths?
We're not talking about abnormalities. Normal people, right, show 8 sort of, and it can interpret 8 emotions, right? And we also have universal facial expressions that match up with these. So you guys, I'm sure, can interpret these. What's this emotion? Of these ones.
Sorry.
You guys know this? What?
Disgust.
Sad?
Fear?
Disgust? Surprise? Annoyed? Right? So, contempt is what that one was. Anyway, we have anger, sadness, happiness, fear. We have these, and we exhibit them, we show them, and we read them, we interpret them. Right? All right, things you need to know about emotion theory. 3 classic theories. The very first theory, and this is definitely something you will see on assessment tests, on GREs, on standardized tests. This is almost always included on that. Changed language theory. This is the original emotional theory. It said that emotion is assigned to a physiological response. So you have a stimulus, you have a perception, you have a physiological response, and then your brain interprets that with an emotion. Right? Then the Cannon-Barth theory said, wait a minute. That's not actually true. Because the physiological response and the emotion doesn't rely on this. Because we get the same physiological response, we just talked about that. Fear and excitement and different emotions. So while we have a stimulus, perception, it splits, and we have an interpretation of that stimulus as an emotion, as well as a body response to that. And those two together create our emotional experience. And then finally, the latest, more modern theory is the Shatner-Singer theory, which says, yes, stimulus, you interpret that stimulus, loud bang, you have your autonomic nervous system. There's a context to that happening. Me hitting the board doesn't elicit much, but if you're walking across campus dark at night and it's dark and someone comes up behind you and claps their hand right behind you, probably that context is going to change your emotional value of that stimulus, right? And your emotional state, in turn, provides feedback that influences your next stimulus, right? So you're walking across campus, everything's fine, it's dark, it's late at night, and you think you hear something. You're like, what was that? So you start listening, you keep walking, and then you have a loud bang. You're going to have a heightened response to then if you're just walking around, everything's normal, and you hear something, and you haven't had that fear to already prime the system, right? So the big difference in these is that the Shatner-Singer inputs that feedback, that influence of emotion on future emotion, current emotion on future emotion. All right, we've already talked about the limbic system. First identified early in the 1930s. These structures are all connected. Cingulate gyrus, why I made you memorize that. Why is that the only gyrus you have to know? that's important with the limbic system. The amygdala, stimulus value, the frontal lobe, all play a role in this circuit. Sort of ancillary things are the fornix, the mammillary device, and hippocampus that all play into this as well as part of the limbic system. All right, if we look at emotion in the brain, there's been a lot of talk about, all right, is emotion lateralized in the brain? Is it like language? Is it found, is happy in one part of it? Is there a happy part of the brain and a sad part of the brain, and an angry part of the brain? Where do these areas come from? And if we look at scans, when people report different emotions, each one of these dots represents areas of the brain that got active for those different emotions. And so the evidence is pretty equivocal, meaning some studies have shown that it looks like language, that emotion could be in certain parts of the brain or hemispheres. Other studies like this show, hey, the right hemisphere is equally active as the left hemisphere for all the different emotions. All right? So look up here, we'll play a game real quick. All right? Which face shows the most emotion? Write down A or B, real quick. All right, which one showed the most emotion? A or B? All right, look over here again. Which face is happy and which face is sad? Write down which face is happy, A or B. All right, last one. Which face is showing the strongest emotion here, A, B, or C? The most emotion shown. All right, what's your answer? All right, so how many people said that A was showing more emotion than B in the first one? Raise your hand. All right, that's a majority. Okay? All right, how many people said that A was showing the most emotion out of those three? Raise your hand. That's less than majority. How many people said B? Okay. How many people said C? Nobody said C? Interesting. All right. Most people say A. How many people said that A was showing happy in that? Most people said A. Right? Majority said A. So what do you notice about these images and these images?
Cut in half.
They're split in half, right? So here the smile is on the left side, here the smile is on the right side. It's the same coming into your brain, right? Same amount of emotion. But most people rule this one as more emotion because we have a tendency to both look at the left side of someone's face to interpret their emotion. Well, this is the right side. To look for our left visual world, right? And here, most people say, again, happy and happy. But if I ask you which one's happy, you look at the left side of the face first, or the left world first, right side of the face first, in order to identify them. Right? Here, this is the normal picture. Nobody said that was showing the most emotion. This is a mirror image of the left side of the face, or no, sorry, this is a mirror image. Mirror image of the right side, right visual world, not right side, it's right visual world. Mirror image of the left visual world, and most people report. So this is like left side and then a flip mirror of the left, right? That this has the most emotion. What's interesting is if we replicate, if you look at the left or the right, both of those you think show more emotion than if we have different levels of left and right emotion here. So studies like that have tried to say that language is lateralized to different sides of the brain. And in general, that the left hemisphere is more dominant for happy emotions, the right hemisphere more dominant for negative emotions. Some of this research also looks at stroke when people have damage to the left or right side, either physical damage or stroke damage. And so people who have damage to the right side of the hemisphere, which side of the hemisphere is looking at left world? Right. And so people with damage to the right frontal lobe have a harder time interpreting language. They show less emotional expression than people with damage to the left. And they have a hard time interpreting emotions, especially subtle emotions. So they may have a hard time understanding irony, or that the tone in someone's voice inflects emotion. They just hear the words, they don't hear how the words are being said, the emotion behind the words being said. Another theory that's a little bit more complicated, but maybe more accurate, says that the left side of the brain may respond to emotion, but it tends to respond to lower levels of emotion, the low and moderate emotion, whereas right side tends to respond to moderate strong. And the left side is more likely to approach based on emotion, like happiness or anger, to know, hey, that's someone I shouldn't approach, or that that's someone I should approach. Whereas, and that would be the activation system, behavioral activation system versus the behavioral inhibition system, which is more of an avoidance, where with stronger things like fear, or not just anger, but like intent to hurt me, danger, that I should avoid and not approach that stimulus, right? So there is some of that. But then when we look at these brain scans, it looks like the areas are equally active. So that's why I say the jury's out, it's equivocal, we're not really sure about this. There's evidence for it. All right, some specific areas, the amygdala. We've talked about the amygdala stimulus value. We also talked about how the amygdala can change its stimulus value, right? So that cheeseburger you used to love after you get the flu, you now hate, right? The significant other you used to love, after a bad breakup, you have different emotional values, right? The amygdala is heavily involved in that. And what we know is, again, primarily from lesion studies and studies where people have their amygdala damaged, which is difficult to do because look where it is. How do you selectively damage that area without damaging a whole lot of other stuff around it? So it's pretty hard to do. But one thing that can damage that is the rabies virus. For some reason, the rabies virus targets the amygdala. Animals that have rabies, what do?
Aggressive.
They thumb them out, they're very aggressive. Are they scared of you? No. Like normally, an animal, a wild animal, would run away from you, right, if you approach them, especially a small cat or dog or something, raccoon. If they have rabies, all that's gone because their amygdala's gone. They don't have fear, they show a lot of aggression, right? And this is because the amygdala is involved with helping regulate those emotions. So originally, because we knew this, the amygdala is thought to be the fear center of the brain. That's not true. It's the emotional value center. It's much more subtle than that. It's just in cases like this, yes, it's clear. Right? This is an example of a person who had selective damage to their amygdala from a tumor, I believe, and they were asked to draw pictures of people that were happy, sad, surprised, angry, disgusted, and afraid. What did they draw for someone that's afraid? Were they pretty good at drawing all the other emotions? I mean, they're no cartoon artists, but I mean, probably better than I could do, maybe. But what did they draw for afraid?
Baby crawling.
An angry looking baby crawling. Do they have a very good emotional representation of what fear is? No. Right? All right. So what do we know about fear in the amygdala? Well, the startle reflex that we just talked about. So write down slide 41. I should probably talk about this in my lab introduction. The startle reflex is a reflex, the acoustic startle reflex. Even babies show this. Right after they're born, you present a loud sound to a baby, it will jump. Muscles will tense. Right? It's not learned. It's instinctive. And we know the pathway, and there's a paper on the website that I want you to use as a reference to cite in your introduction. You'll get points off if you don't cite it correctly. But it's a big paper, and I don't want you to waste a lot of time. So I've told you what sections are important. So you can ignore all the other sections, but look at the specific sections on the website that I say, hey, these are the ones that are important, right? And in there's a diagram, there's a figure that's important. You're writing all this down, right? You wanted to go home your lab report. That identifies the pathway from the cochlear nucleus to the pons, to the red nucleus, to some other nuclei that we know is automatically triggered to cause a loud sound to cause your muscles to tense up in response to that acoustic startle reflex. Right? If we inject lidocaine, an anesthetic, to block activity temporarily in the amygdala, we eliminate the ability to have sensitization to that startle reflex. So those of you that had sensitization to the screen, guess what? Your amygdala went, wow, be afraid. And the next acoustic startle zone, you had a bigger response. If we inject CCK, that's a neurotransmitter that stimulates the amygdala, and we don't even have to give the human screen. On the next tone, you will, for no known reason of your own, have a bigger response because we've activated your amygdala artificially. All right, we're getting there, guys. We're getting there. So the amygdala is involved with emotional value for specific items. There's another area of the brain called the bed nucleus of stria terminalis, and this is located here. It also reaches around and connects with the amygdala. But basically, you can think about this as a forebrain area. And this area is sort of involved with your overall level of anxiety or non-anxiety alert, right? Sort of long-term adjustments for anxiety or fear. And it could be that people that are highly anxious, that this area is more active than other people. I know that for sure, but it's a good indicator. And then there's other living structures that are involved with fear and emotion as well. But these are two of the big ones. The amygdala is sort of regulator of the whole system through the bed of stria terminalis. A little bit about aggression. Aggression also involved with the amygdala. So famous study, Kluver and Busey were researchers, I forget if they were at Yale or Harvard, one of the Yale or Harvard. They were one or the other, and they were working with primates. Everybody's like, oh, it'll be so fun to work with monkeys. No. Monkeys can be very aggressive. They throw their poop at you. They want to bite you. You can catch diseases from them. It's like sometimes very difficult to do monkey research. And they found out in the early 1800s that, hey, if we go in and take out their temporal lobes, they become much easier to work with. Maybe not if you're trying to do learning and memory, since that's where learning and memory is located, but for what they were trying to do, it calmed the monkeys down a lot, right? And this was a person, but what they really were doing was taking out their amygdala and areas that are involved with the limbic system. So anger or aggression, right, once it's triggered, can last for a pretty long time. And it can be transferred to other people and objects, right? So why does this happen? What happens when we become angry? You're driving your car and some jerk pulls out in front of you, right in front of you. Almost kills you, right? What do? Right? Your autonomic, your sympathetic nervous system physiologically gets activated, but what do you do behaviorally? Some specific gestures, some specific words. There's some anger and aggression, right? They don't see you, they drive off, they're gone, right? And then your roommate is riding with you, reaches over and changes the radio station, and you yell at him. Because the sympathetic nervous system is still activated, and you're now transferring that anger for the jerk that almost killed you to your poor roommate that just changed the radio station, right? And it takes time for the parasympathetic nervous system to change that arousal state, right? The ability to become aggressive and angry is associated with levels of testosterone, particularly in men, both acute levels of testosterone and chronic levels of testosterone. So we know that testosterone can act on neurons in the hypothalamus to produce an increase in aggressive behavior. If we look at the level, there's been studies looking at inmates and they're circulating chronic levels of testosterone. Inmates that are convicted of violent crimes like rape and murder have a much higher percent of those inmates have high circulating levels of testosterone than inmates that are convicted of that are serving in the same prison but convicted of non-violent behavior like drug offenses and burglary. Much smaller percent show high levels of testosterone. So there's lots of studies that have looked at, does testosterone influence your level of aggression and anger? Here, if we look at animals, males tend to bite, male rats tend to bite more than female rats. How many of you ever took, and you all never took learning at that today, Dr. Reed, because you and I know that course. Dr. Reed did a lot of experiments with handling the animals, training them in these rigs to push levers and everything. He always used female rats. Always, because they were less likely to bite than male rats. If we take male rats here's female rats, much less likely to bite. Which rat would you rather work with? Female rat, right? Unless you like getting bit by rats, right? If we take a rat and we castrate them, we remove their testes, therefore no more testosterone, look what happens to their biting behavior. So obviously testosterone, big influence. If we remove the ovaries of a female rat as a control, nothing happens, right?
So is that why people, like, when they have dogs and stuff like that, they neuter their dogs so they can calm down and be less aggressive?
Mm-hmm. Sure. Dogs will be less aggressive if they have less testosterone. And that's probably not why they neuter them. They usually neuter them so they can't pregnate a dead dog. But it does have that effect as well. And so here's a rat, castrated, no testosterone. Hey, let's give it injections of testosterone. And guess what? As we give it more testosterone, the biting goes back up. We take away the testosterone, the biting goes back down. It's a pretty clear relationship between the presence of testosterone and aggression. All right? It not only can cause aggression, it also changes our perceptions of aggression. And so here, human subjects were asked, so this is a non-aggressive face. We'd agree that's an aggressive face, right? In between are increasing levels of aggressiveness. People were shown these slides after either being given a placebo injection or an injection of testosterone. And when they were given testosterone, they had a 10% more change in where they said they saw aggression. So it took them 10% more emotion, per se, right, to recognize aggression under the influence of testosterone than people without. So not only does it make you more aggressive, it makes you less in tune to detecting aggression. So both of those things can lead to what? More conflict. Like you are more aggressive and you don't recognize the person you're being aggressive to as being aggressive back. Leads to conflict, right? All right. Final thing, I promise, well, next to final thing. Next to final thing. Serotonin also plays a role in this. And it turns out that we can measure serotonin in the brain by looking at its metabolite. So how does serotonin get removed from the brain? Do you remember? Primary ways serotonin gets removed from the brain? From the synapse and so on. Reuptake back into the cell, and what else?
Is it Gila cell or Gila cells?
Glial cells. Good job. Release what? You're on it. They release enzymes that are going to break it down, right? So we have MAOs, monoamine oxidase, that breaks down serotonin, good, functional glial cells, and reuptake, two mechanisms. When serotonin is broken down, it's turned into this metabolite called 5-HIAA, and then the glial cells dump that into blood. So we can measure circulating levels of this, and if you have low circulating levels of this, what does that mean? What does that mean about this? Low levels of this in the brain, right? And this is what low serotonin turnover when people talk about that, what that means. Lower levels of the metabolite in monkeys and humans is correlated with more violent behavior, and in people that are depressed that have low levels, the people that have the lowest levels are more susceptible to attempting suicide, and those that have attempted suicide multiple times have even the lowest levels, right? So seasonal suicide is also related to low levels of serotonin. Suicide being a violent action against oneself, right? And so somehow serotonin not only can underlie depression, but can also underlie the propensity for violence as well, along with testosterone. So these things, again, they're complicated. It's not like there's just testosterone as the aggression hormone. No, serotonin plays a role in it as well. There are interactions between the environment and this. So here we have, in a normal population, we're going to talk about childhood maltreatment, so children that were abused. None. Children not abused, our control population. When we have high monoamine oxidase activity, there's a gene that codes for this. So people can have high amounts of this enzyme or low amounts. If we have high amounts of this enzyme, what does that mean to our levels of serotonin? They're lower because we're breaking it down more effectively than here. And they tend to have higher antisocial tendencies, violence, than these children, right? Now, here's where it gets interesting. If there's probable or we know there was severe child abuse, it influences this propensity. There's an epigenetic change in interaction between that child's environmental experience and their genetic background to be high breaking down serotonin or low breaking down serotonin that actually flip-flops that. So that now the ones that actually don't break down serotonin, because of the influence of that maltreatment, are showing more violence than the ones that can break it. So again, it's complex. It's not strictly physiological. There's a lot of environmental changes, right? The gene for this is found on the X chromosome. So again, it's sex dependent. It affects men more than women because women have 2 copies of the X chromosome. Back to our reproduction. Now, the final thing, I promise this is it. We'll be done before 4:30. Right here is the lateral hypothalamus. What does the lateral hypothalamus do? Eating, feeding. If we stimulate the lateral hypothalamus, what happens? You eat more. You just act like you're eating. If we inhibit it, what happens? Animals what? Or we damage it. They stop eating, right? Scientists were sticking electrodes down in the lateral hypothalamus and either stimulating it or inhibiting it, and looking at animals pushing a lever, like how hard would they work for food? If we stimulate this, guess what? They work really hard for food because they're hungry. And if we inhibit it, they quit working for food. They don't work as hard for food because they're not as hungry, right? Scientists stuck an animal in there, started inhibiting this area, and what did they expect to happen? Animal won't work hard for food, not hungry, right? Follow me? They start inhibiting the area, the animal starts pressing the bar harder than they've ever seen it press the bar, as fast as they've ever seen a rat press the bar. Food pellets are just flying out of the hopper, piled up in the corner. The animal doesn't even care that there's food there. It just keeps jamming on this bar. Turns out they missed the hypothalamus with their electrode, and they hit this area called the nucleus of accumbens that's right in front of it. So they're like, what's going on? So they sacrifice the animal, look at where the electrode was. Hey, we didn't go to the right place. That animal would bar press 2,000 times an hour until it passed out from exhaustion and fell over with all the food piled up in the corner. And then when it woke up, it would jump right back on the lever and start pressing it again. They had found the pleasure center, the reward center of the brain. When I say there's not a love center of the brain, there is a reward center of the brain. It's called the nucleus of accumbens. So when you inhibit the nucleus of accumbens, you feel intense joy, intense pleasure, ecstasy. When you have your most ecstatic emotional moments, your dopamine is greatly inhibiting the D2 receptors, the nucleus of accumbens. This is an example, and it's really short.
Perhaps to get pleasure, because they stimulate the same areas as sex.
And really, all I want to show you is, here's an animal. I just want to show you how this really works. Lever press, presses that lever, you'll see the little light come on. This is back in the 50s when people first learned this, right? Presses it, gets inhibited. He's like, oh, I like that. And he's just immediately jumping on it, pressing that lever, self-stimulating, inhibiting the nucleus of accumbens. The interesting thing about this is that not only will they press the lever, they now train it that you can press the lever on this side of the cage, but after 5 or 10 presses, it quits working, and you got to run to the other side, and now you can press that lever, and it'll stimulate. And then it quits working, and you have to keep going back and forth, running back and forth. They electrify the floor here, so that the rat gets an intense shock when it runs across the floor, and it will tolerate running across the electric shock because the reward is so much greater than the pain of running across the electric floor.
Brain the shock is stronger, and even a starting rat brain together.
So the point I'm showing you
Human addicts will take just as high a price.
is what he's just saying is that drugs of addiction, all drugs of addiction in one way or another, act on the nucleus of accumbens to provide that reward. When you use that drug and feel good from using that drug, it's activity in the nucleus of accumbens. And addicts, once there's a change, and we'll talk about this in exam 4 when we talk about addiction. We're talking about it here because of emotion and pleasure. But once that change happens, addicts will do ridiculously dangerous, self-endangering behavior that no normal person would do in order to get that drug, for the same reason that this rat will run across this electric grid, because the nucleus of accumbens has been fundamentally changed in their brain because of that drug use. And it really explains some of the abnormal, destructive behavior that addicts show based on this physiological change. All right. The dopamine that's activating the nucleus accumbens comes from the ventral tegmental area back here. And so some drugs act through the ventral tegmental area to cause it to release a lot of dopamine in the nucleus accumbens. Nicotine, one of the most addictive drugs, people that are addicted to heroin and nicotine report that it's harder to get off of nicotine than it is to get off of heroin. Nicotine acts here to stimulate the VTA, the ventral tegmental area, to release dopamine. It doesn't act directly on the nucleus accumbens, but it acts through there to cause that huge dopamine release. Why do you think the lateral hypothalamus and the substantia nigra are shown on this slide? What does the substantia nigra do? We already talked about it. Releases dopamine, but where does it release dopamine? In the basal ganglia. It goes to the basal ganglia. It doesn't go to the reward center. right? The VTA goes to the reward center. So it's a different dopamine center to a different part of the brain for a different job. The lateral hypothalamus actually, remember we talked about reward and I'm so worn out, I can't even remember my thoughts either. We're talking about comfort foods, right? And we talked about eating for comfort. Yes, our lateral hypothalamus actually has a role into that reward system. When people talk about becoming addicted to foods, because there can be that association that can occur in that same circle. All right. So dopamine activates, dopamine inhibits pleasure center. Glutamate excites, brings it sort of back to normal or non-reward. We don't always want to be rewarded. We only want to be rewarded when good things happen. When bad things happen, we don't want to be rewarded, right? So these things act in opposition. And as I mentioned, drugs like cocaine cause dopamine release, but other drugs like PCP block glutamate. And if we block glutamate here, we get the same effect of inhibiting. So either an antagonist to glutamate or an agonist to dopamine can be an addictive drug. And this summarizes some of that stuff. All right. You guys are good sports. I'll see you 9:30 tomorrow. We'll talk about stress. Make sure you record your ASR data into the group data sheet before next Wednesday morning, 8 o'clock. And no time like the present to do that. And your method and intro due start class Tuesday. I'll see you